Thanks very much for the introduction. I feel a little bit like a fish out of water here. I'm a nephrologist by background, and here I am talking to a bunch of people who know much more about brains than I do. So I'm going to talk to you about something completely different, which should keep us all on safe ground, which was the recovery trial. And I'll try and explain why I think that might be relevant to people in this room as we go through. I hope it doesn't put too many people into shock, but just to take you back to March 2020 and the situation that was evolving in the UK at the time with COVID-19, is that we had a clear rationale for randomisation, and I think this is one way in which the UK led the world, in that our approach to this major public health crisis, where people were going into a hospital at an alarming rate and not coming out of hospital at an even more alarming rate, with huge uncertainty about how to manage these people, the UK decided to adopt the approach of randomisation wherever possible, both in the recovery trial and other trials that were going on at the time. There were many different drugs that had been suggested. There were almost as many opinions from varying sources, including some very loud political opinions on what should be done, but there were no reliable data on which to guide treatment. And elsewhere in the world, opinion seemed to take precedence, and people were treated as people thought best. But in the UK, we managed to get a lot of support from Chief Medical Officer down for randomisation. And randomisation was really essential because we knew there was unlikely to be a single drug that was going to cure this disease, no big win, and therefore we needed to find moderate treatment effects. And if you want to find moderate treatment effects, whether you're treating COVID-19 or dementia, then you need large numbers of people and you need randomisation. And so that was the approach that we adopted. Unfortunately, it was done at a time when randomised trials themselves were in a bit of a crisis. It's undoubtedly, over the last 20, 30 years or so, there's been an adverse impact over unnecessary regulation and bureaucracy on randomised trials and the way that they're conducted. As I'm sure people in the room who work on them know, there are increasing numbers of obstacles put in the way when you try and set up trials, which drive up the costs and effectively drive research towards cheaper, quicker ways of getting answers, but unfortunately methods of developing evidence which may not be or are not as reliable as randomised trials. And the research agenda therefore gets distorted, and innovation dies in the face of overwhelming bureaucracy, which is a tragedy when we've got so many diseases, so much exciting science, basic science, giving us potential treatments that we can't take more of them through to randomised trials. And ultimately, trials become a matter of trying to obey the rules rather than innovating and trying to come up with right answers which can improve public health. So we had a public health crisis, but we also had trials in crisis at the time. But that gave us an opportunity when COVID-19 came along to actually think about how we really wanted to do this and how we were going to address it. And the way we did that was not by coming up with some new idea, some new way of doing trials, but it was actually to look back, and in particular to look back on a trial called the ISIS-2 trial, run in the 1980s, in people admitted to hospital with myocardial infarction. This had been run by some of my predecessors at where I work in Oxford. But with this very simple and very elegant trial, they found two treatments which reduced the mortality from acute myocardial infarction from 13% to 8%. And so those results changed practice overnight and have saved millions of lives since. But what they knew at the time was that to make that trial a success, was that it had to have the minimum impact on the usual care process that the patient was going through at the time. Any extra work required for the trial must be, and by design of their trial was, absolutely minimal. And that allowed them to recruit the nearly 18,000 people they put into that trial to develop that evidence. But those two things are linked, that if you want to develop really reliable evidence, you have to think about how your trial design and make it as streamlined as possible, and that's something I'll come back to. So randomised trials don't have to be complicated, but they do have to be practical. So for COVID-19, that meant that we took a very simple approach. People were eligible for the trial as long as they'd been admitted to hospital and they had suspected or proven COVID-19. We chose a very easy outcome to measure, mortality, unfortunately very common, but easy to measure at least. And then we did have some secondary outcomes. We kept them relatively few, and also objective and simple to capture for sites. Did the patient need a ventilator? How long did they stay in hospital? Crucially, there was randomization at the heart of it. Randomization guided simply by a local doctor deciding which of the available treatments the patient may be suitable to receive and were available in their hospital. And as long as both of those things were true, then those drugs were included in the randomization. And then we kept follow-up as simple as we could. So we had a one-page case report form to be completed when the patient was discharged or died. And then, as I'll tell you more about, we had a substantial amount of linkage to national data sets. We tried to make this as easy as possible for sites. There were a lot of uncertainties at the start of COVID-19. We didn't really know how things were going to work. We had a paper consent form to get things going as quickly as possible, just a one-page consent form. We then discovered that, unfortunately, many infection control practices at sites meant that those consent forms were burnt when the patient left the ward, which creates a bit of a problem for a trial. So we had to come up with some ways to get around that. We had a one-page electronic case report form to be completed to randomize the patient. So that we measured that. That took people about 3 or 4 minutes to complete that. And then the randomization was as shown on this diagram. Initially, people were randomized between one of 5 different arms. Everybody got the best standard of care that was available in their hospital. And then on top of that, they either got nothing else or they got 4 drugs which had been picked for us by a national committee as being appropriate drugs to test if there was a respiratory virus pandemic. So those are the ones we tested with those outcomes. So we kept this as streamlined and as simple as possible as we could for the hospitals. And in terms of recruiting patients, we then took to the trial to them. So you can see at the map on the left, each of those green circles is one of the hospitals that took part in the trial. The size of the circle is proportional to the number of participants. You probably can't see it. There's a little green dot on the Western Isles just off Scotland at the top left, but we covered the UK. We got every acute hospital taking part. And some of these hospitals did, or many of these hospitals did fantastically well. If you look at where the big green circles are, some are based in academic teaching centres, but many are not. There were hospitals which didn't have much history of taking part in research up to this point who embraced this trial, who's from the board down, they decided they were going to make sure that recovery was a success, gave the teams the resources they needed, and so some of our largest sites were hospitals that probably haven't featured in many publications before. But it allowed us to recruit very rapidly. So you can see the recruitment curve on the right-hand side there. During the first wave, in the spring of 2020, we recruited 10,000 people in the first 8 weeks, and that was largely what led us to the first round of results that we had. We all then enjoyed a bit of a summer off. COVID went away temporarily, but then it returned with a vengeance in the next winter that you'll remember. And each of those vertical lines you can see there on the graph shows the recruitment of 5,000 people. And at one point in January 2021, we were recruiting 1,000 people in about 30 hours. So the NHS poured huge resources into this, but it meant that we got hugely reliable answers out, for which we're all very grateful. There were 2 problems that were vexing us when we started the trials. One, would we be able to recruit people? Could we actually get some order into what was becoming an increasingly chaotic NHS? And there the NHS did us proud and recruited very well, as I've just shown you. The second thing was, it's all very well recruiting people, but will we then be able to get the outcome data back? It's no good randomizing people and then just losing track of them. That would be no use to anybody. So we put a lot of effort into leveraging routinely collected data, which is another huge strength of the NHS. There are large numbers of data sets, more than I imagine, more data sets than I think UK Biobank is currently linked with, available for us to use in recovery. Data sets around hospitalisation, critical care, COVID-specific data sets, primary care data sets that we linked our participants with. And actually that allowed us to collect pretty much all of the outcome data that we wanted without even having to ask the sites. We did ask the sites as well. We tried to get complete coverage in both ways. And we've had some fun looking at the results of these trials, whether you just use the linkage data or whether you just use the site data. The reassuring answer is you get pretty much exactly the same answer, whichever whichever data set you decide to look at. But this meant that we had a backup in case things went wrong, and has been a real strength. It also allows us to do very long-term follow-up very, very cheaply indeed. So putting those a streamlined protocol together, fantastic effort from the NHS, in particular NIHR staff embedded within it for recruitment and for providing follow-up data. It's allowed us to identify 4 different effective treatments for COVID-19, all of which feature in guidelines nowadays. Cornelia said I might talk about stratification. I'm not going to talk much about that, but I think many of these results do show the benefit of recruiting broadly and then stratifying later. For example, the dexamethasone result, the first flagship result, if you like, from recovery, showing that dexamethasone can reduce mortality. The benefits of dexamethasone varied greatly depending on how severely ill somebody was, such that patients who weren't particularly unwell, who'd been admitted to hospital but didn't need oxygen, there was actually a suggestion in our results they may be harmed by dexamethasone. So those patients now don't receive it. But if we hadn't studied everybody, we may not have been able to make that important distinction. Happily, the sicker you are, the more benefit you get from dexamethasone. But then we've added tocilizumab, Ronapreve, which was the first drug specific for COVID-19 that we got into the protocol. This is a monoclonal antibody combination directed against the spike protein. We found that was beneficial, reduced mortality by 1/5 in people who didn't have their own antibodies at baseline. And then more recently, baricitinib, showing that we could weaken people's immune system more than anybody would have dared reasonably possible at the start and still yield mortality benefits. Equally importantly, we've discovered a lot of treatments that are ineffective, and many of these treatments had very strong advocates. Hydroxychloroquine had some strong advocates from North and South America, from presidents saying that it must be used in the treatment. It's the one treatment we found that we stopped looking at it when it was looking like we were going to find clear evidence of harm. We could abandon it and get on with other more useful treatments. Azithromycin, a commonly used antibiotic with lots of people thinking it could have antiviral properties. We found no benefit there. One example I'll draw your attention to is convalescent plasma. I know you can't see all the detail of these, but I think you can see that the red line and the black line are on top of each other, which is all you really need to see. Convalescent plasma in the US, they had a big convalescent plasma program. This is where you collect plasma from people who've recovered from COVID and then use it to infuse into people with a fresher diagnosis to see if you can give their immune system a bit of a helping hand with some antibodies. In the UK, we were lucky to work with NHSBT, who selected plasma, the most enriched plasma that we could get, so they had high titer antibodies. In the US, they did a similar program, and they spent about $100 million on giving convalescent plasma to people, but the one thing they never did was randomize people. So they've produced a lot of evidence suggesting that it might work, and there's some strong advocates over there that convalescent plasma is a useful thing to do. In the UK, we randomized about 11,000 people and showed that it had no benefit at all in a randomized trial, suggesting that those effects that they're seeing in the US are very likely due to bias, due to patient selection. And so this has allowed many efforts to be refined. Convalescent plasma still may yet have a role, collected in different ways, used in specific types of people, but in terms of a broad measure of treatment for most people, I think we can reliably say it's not effective. So I talk a lot about streamlining, about keeping things simple. Sometimes people think that equates to a low-quality trial, that you're losing, in some ways, some power of the trial. I would argue to the contrary, that if we can streamline trials and thoughtfully apply quality by design principles, in other words, deciding what really matters for your trial and focusing on that, and trying to strip away things which some people might want because they think they might want to look at it one day, but just focusing on generating really high-quality data while looking after patients, you can get very reliable answers, and I think recovery is a great example of that. If you don't want to take my word for it, this is an article from JAMA Internal Medicine, written by the current commissioner of the US FDA and 2 people very high up in FDA underneath him, talking about the recovery trial and its application in the US. And a key point they make is that I highlighted here is that streamlining and quality are not opposed. That you can have a very high quality trial and it can yet be very streamlined, and then you get very, very reliable answers. In COVID-19, unfortunately, I don't think COVID-19 is unique, but it's a defined disease that we can look at. Unfortunately, too many trials fail by design. This is, again, more data generated by the FDA. Looking at all the trials registered on clinicaltrials.gov, only about 5% of the nearly 3,000 comparisons that have been registered on clinicaltrials.gov were into comparisons that were adequately powered, that had any chance or realistic chance of detecting the treatment effect that they set out to. So therefore, 95% of trials were very, very likely to fail. Of the just over 500,000 people recruited into those trials, only about 1/4 were recruited into the 5% that stood a chance. So in other words, 3/4 of the recruitment effort was wasted. And patients were going into trials that really stood no prospect of doing them any benefit. And that's partly because of the crisis that I talked about earlier, that people were designing trials too burdened by regulation and therefore couldn't reach the size that they needed to be. In recovery, we tried to break that paradigm and show that you could get quality and yield good answers. So what makes a good trial then? Well, in my opinion, you need to ask an important question. I put important in quotation marks there because I think that's a slightly subjective opinion, but broadly speaking, it's testing a practicable treatment in a common condition on a major clinical outcome. Once you've picked your question, then answer it reliably. And if you want to do that, you need to randomize people properly. Don't introduce any bias into the randomization process, and don't introduce any bias into the follow-up process either. And randomize sufficient numbers of people so that you can minimize your random errors in the experiment as well. And focus on the safety of patients. This is ultimately what GCP guidelines come down to, is safety of patients. The safety of the patients going into your trial itself, and the safety of future patients whose treatment is going to be affected by the trial that you deliver. In other words, generate reliable data so that future patients are treated in the right way. I'll just divert for a moment. As I say, I'm a nephrologist by background. I was in clinic this morning, and when I was in clinic, these are the sorts of issues that bother me in clinic and in the trials that I try and run in kidney disease, is that we have multiple rare diseases in kidney disease. We have some common ones, diabetes being a good example, but many of these common diseases end up in a common clinical phenotype. This is people with impaired kidney function, which then deteriorates over time. There is superimposed on those diseases a ubiquitous age-related decline in kidney function, but these other diseases accelerate it, and in some people accelerate it to the point that their kidneys may fail them before they die of something else. In many people, this disease progresses in a fairly linear fashion, a fairly predictable fashion, but in some people, there are acute step changes superimposed on that, often associated with other acute illnesses, but these can accelerate the rate of decline. In nephrology, we have a continuous measure of function. It's not perfect, but as far as trials go, it doesn't need to be perfect. We don't need to measure precisely what somebody's kidney function is in one arm of the trial. As long as we do the measurement in the same way in the other arm of the trial, then the sorts of errors that leak in a way cancel each other out, and what we can focus on is the randomized difference. So we don't need a perfect measure. We do have some clinically defined hard endpoints. That's when people start dialysis. But when people start dialysis itself is quite variable. There's a lot of variation in practice about when that's done and how that's done. But again, in a randomized trial, that doesn't matter. And we have death as a major competing risk. Most people with kidney disease actually die of something before their kidneys fail them. But it's an important consideration with trials. And the reason I run through that is that when I think about dementia, I actually think you could probably say exactly the same thing about dementia. And in nephrology, we are now, having been at the bottom of the league table for trials, we're making progress. We are running large, streamlined trials and finding treatments which improve outcomes. And I think many of the lessons that we've learned in nephrology about streamlining trials could probably be applied in some way to dementia trials. So what do I see as trials of the future then? Well, they need to be designed cleverly and thoughtfully, not done following a template, but thinking about the problem and designing a protocol that addresses it. If possible, integrate it with routine healthcare. There's huge benefits there in terms of completeness of follow-up, additional follow-up, generally at minimal cost and intrusion to the participant themselves. We need to make sure that these regulations, which have blighted trials over the past 20 years, are proportionate, and there are efforts going on led by Wellcome and Bill Gates and the Bill and Melinda Gates Foundation to try and improve trial regulation and guidance. And as academics, we need to get behind that and make sure that the trial regulations are scientific in their origin rather than bureaucratic. And we can do all of this for the benefit of better patient care and improving public health. So I'll finish there. I've talked about recovery. I do need to thank, we had 175 sites, all with many, many staff, which we need to say thank to, but I always like to finish by thanking the patients who at a terrible time in their lives took a chance, let themselves be randomized, and have helped us generate the data we have. But thank you for your attention.
Coote, there we are, Brandy Coote, I now work with Recovery Health, but worked in the NHS for over a decade and set up many of the COVID trials in addition to recovery in Oxford. So first, a comment for you. I think it's absolutely, I've never seen it replicated before or since, obviously, the 175 sites to be able to open that quickly as you guys did. Having worked with your team, you guys did a brilliant job, not just in your smart design, but that delivery piece, and that was really crucial, I think, to getting the studies up and running and to get that kind of uptake from trial sites. We know and we recognize that it was a very unique environment, obviously, at that time, and everyone was there to do exactly those types of trials at exactly that type of time, and I think that aligned really well. But absolutely credit to you and your team, because I think you guys did a brilliant job. My question to you is in relation to the importance that data played and is currently now playing in your long-term follow-up for those participants. Do you still have the same access to that data now as you did during that time? Because of course we know that some of those, that openness, that initial openness has since ceased. So do you still have that, and what are your thoughts on that continuing into the future for the benefit of all trials across the life course?
Yeah, you're right. There were some data sets created just for COVID, which we got access to. But actually, all of our data access is on the basis of we have consent for it all from the participants. So in fact, the fact that some of the directives on access that may now have ceased, that doesn't affect us. We still have the same rights to access it. So yes, we do. All of those data sets are still flowing. Not as frequently as they were. We were getting weekly updates at one point from the data custodians. Now we get them every 6 months or so. But no, for the long-term follow-up, we will be secure, and those data will be secure. And that wasn't new for recovery. We've done that sort of linkage before and secured it for 10, 20 years after the trial is finished.
Really great talk, thank you. Just the kind of learning we need. Did you explore the patient's perspective at all? Did they know, did they really understand that they were in a trial, or that they had been randomized, or that they thought they were making an important contribution? What did the patients think about this?
Good point. We actually did get a question when we first were trying to set the trial up about what patient and participant involvement we'd had, and obviously at that point it was almost impossible because there was nobody with COVID to talk to. We have set up a participant panel which meets fairly regularly to talk to them about the trial and our plans, and they give us useful input on them. And one thing we have done is written. We've got now just over 48,000 participants in the trial. We've written to them all twice to tell them about the trial and tell them about the results. So they know that they're in a trial, they know that they've contributed to it, and they have an opportunity to communicate with us back if they have any questions. Of all of those 48,000 people that we've gone into, less than 10 have subsequently raised a concern they didn't realise they were in a trial. in the first place, which I think is probably credits to the people who took their consent in the first place. But no, we have made stringent efforts to keep in touch with those participants.
Got time for one more quick question for Richard.
Did you verify that the patient on different drugs were at the same level of severity in the disease?
No, we didn't verify that everybody had the same severity of disease. We accepted people with a broad range of severity. We characterized that severity at baseline in everybody. And then when we did our analyses, we could see that they were well balanced between the two treatment groups. So it didn't matter, but we did a few subgroup analyses to see where the treatment effects were modified by how severely ill people were. Really, the only case where that was the case was dexamethasone, which was fairly unusual. Nearly all the other treatments were pretty constant in their effects, no matter what the characteristic you're interested in of the patient.
Ivan, if you're quick.
Ivan gets another question.
Very, very quick. Richard, I guess the thing that I'm wondering with this sort of complex trial where you have a number of different interventions which differ a lot in terms of what their biological mechanisms are, when you're randomizing people, how are you dealing with this complexity of different mechanisms of actions in people who are very unwell on a number of other medications, number of comorbidities? How was that managed?
Essentially, we let the person in charge of the randomization, the recruiting doctor, make the decision which comparisons they wanted that patient to go into. So if they were clear in their mind that a certain drug was definitely the wrong thing to do or definitely the right thing to do and therefore should just be prescribed, they didn't put them into that comparison. But otherwise, they let us roll the dice. And then all of that complexity goes away with randomization. If the patient went into the control group, then they could contribute to multiple comparisons, but otherwise they just went into one of the arms, and it didn't really matter how complicated the case was. That's one of the beauties of randomization.
Thank you very much. One last thank you for Richard.